The genetic code is the nearly universal set of rules that determine how genetic material can program a specific protein structure. The experiment discussed in this video shows how the nucleic acid RNA can code for a particular amino acid, which are the building blocks of proteins. There are 4 nitrogenous bases in RNA and 20 amino acids. The scientist, Francis Crick, hypothesized a non-overlapping code so protein sequences would not be restricted, and it was determined that there would need to be a 3-letter codon that would code for each amino acid. But how would you figure out what codes for what? The researchers, Marshall Nirenberg, Henrik Matthaei, and Philip Leiter, sought to figure this out. In 1961, Nirenberg and Matthaei conducted an experiment where they mixed together poly-U messenger RNA, this is essentially a chain of uracil nucleotides, with the 19 amino acids and 1 radioactively labeled amino acid in a cell-free translation system. 20 test tubes were made for each amino acid to get radioactively labeled. After filtration, the protein produced in each test tube was tested for radioactivity. The scientists observed radioactivity in the test tube that contained radioactive phenylalanine and proved that the codon UUU codes for the amino acid phenylalanine. In this experiment, they also identified the codons for proline and lysine using the same technique with different homopolymers such as poly-C and poly-A. In 1964, Nirenberg continued deciphering the genetic code with Philip Leiter using a modified experimental design. Short messenger RNA with known codons were mixed with ribosomes and 20 transfer RNA with each amino acid bound. One of the transfer RNAs had a radioactively labeled amino acid attached. The ribosome would then bind with the transfer RNAs that have the amino acids the messenger RNA coded for. This solution is filtered, leaving only the transfer RNA molecules bound to the ribosomes. If the filter contained radioactivity, the labeled amino acid had been coded for by the codon in the short messenger RNA. Through this method, Nirenberg and Leiter were able to identify the remaining 61 codons and which amino acids they code for. Thanks to these experiments, we have access to a dictionary that teaches us the language of DNA. Isn't that cool?